Hello and welcome to my video about DNA to protein. In this video, what we'll be talking about is how we go from our DNA inside our nucleus to a protein. Now there's two processes, transcription and translation. Each process has three parts. One, initiation. Two, elongation. And three, termination. This is basically your start, building up your chain, and your end. So, initiation. Now, initiation, what happens in this is RNA polymerase is the enzyme which is used to make your mRNA strand. And what it does is it looks for recognition sites. When it finds a recognition site on your template strand of your DNA, it will start to read that strand from the 3' end to the 5' end. So, this part here is the transcribed region. It will continue reading until it comes to a stop, but we'll talk more about that later. But basically, your initiation is where it's looking for these recognition sites. It joins on there and starts to read. So, as the RNA polymerase starts to read your template strand, it will start to add on RNA bases, complementary RNA bases, with uracil instead of thymine. The reason for that is uracil isn't as stable as thymine, but it's a bit quicker to make. It's smaller, which makes it a bit easier to manage for the short amount of time that RNA is actually present. Now, it will read from the 3' end to the 5' end, as I said earlier, on a template strand, which basically means your RNA, your mRNA, is the same as your non-template strand, except you have uracil rather than thymine. So, after initiation and elongation, we now come to termination. So, termination is a bunch of bases which basically tell the RNA polymerase to stop reading. In this case here, we have the hairpin. It's an example of something which tells the RNA polymerase to stop reading. Now our mRNA strand is nearly ready. Before our mRNA strand is ready to be translated, we have to remove certain parts of the non-coding DNA. These are called introns. So a splicing mechanism happens, which you don't need to worry about right now. But basically, the introns are spliced out. The exons are then joined back together with a 5 cap and a poly A tail. This makes your mature RNA. Now, your mature RNA is now ready to be taken to the ribosomes and be translated. So, in translation, what happens is we've gone from a two-stranded DNA, double-stranded DNA, to a single-stranded mRNA. We are now in the ribosomes to be made into a polypeptide chain of protein. So, before we move on, we really need to understand this chart. What this chart basically shows you is what codon will, one triplet codon will code for which amino acid. So, for example, UUU will be phenylalanine. Now, what the initiation stage of translation is where this MET codon is looked for, methionine. Every single polypeptide chain will start with methionine. This tells the tRNA this is where this mRNA starts. You need to start reading here. There are loads of tRNA molecules and they all have different amino acids. They all contain different amino acids. So basically, think of them as bridges between your mRNA to your amino acid chain, your polypeptide chain. So what will happen is, as we said before, initiation with the AUG, methionine, then the next codon is exposed. After that next codon is exposed, another tRNA molecule comes and joins. So this process is elongation. So what's happened, we've initiated with our methionine having joined, tRNA from methionine having joined the mRNA chain. Then the next polypeptide linked to a new amino acid is joined together. mRNA shifted forward, exposing new codon, and the process repeats. And this keeps happening until a termination happens. So termination is when the part of the mRNA is reached which codes for a stop codon. That's UAA, UAG, UGA. At this point now, your polypeptide chain is complete. So the tRNA molecules no longer read that bit of mRNA till another methionine is reached, which means to start the process again. So we now have our completed polypeptide chain, which is now our primary structure of proteins. Then it will fold over, as we've done in lessons before, it will fold over in our secondary structure, which is your alpha helixes and your beta pleated sheets, and your tertiary structure, which are your interactions between different amino acids, such as van der Waals, ionic forces, etc., etc. And you may also interact with another polypeptide chain for your quaternary structure. That is how we go from the DNA molecule to proteins. I hope this video was helpful. Please leave me any feedback to let me know how I can improve it. And thank you for listening. Thank you.